Hello and welcome back to Continuity of Life. I'm Emily Hayes and today's episode is called Chains Unbroken. We're going to unravel how life keeps going, across generations, across species, and, well, across time itself. So, let's start at the very beginning with the genetic highways that connect us all. Now, DNA, deoxyribonucleic acid, if you want to be fancy, is basically the master blueprint for every living thing. It's like, I don't know, the ultimate instruction manual, but with way fewer missing pages than the ones you get with flat pack furniture. DNA's job is to make sure that when life reproduces, the instructions are copied with astonishing accuracy. I mean, the error rate is something like one mistake in 1 billion nucleotides, thanks to these proofreading enzymes, DNA, polymerase, and all that. It's wild to think about, really. That's how you get continuity, but also just enough mistakes, mutations, for evolution to do its thing. And when we talk about inheritance, Mendel's P's come to mind. Dominant and recessive alleles, the whole Punnett square business. But it's not just black and white, is it? There's this whole layer of epigenetics where chemical tags on DNA can switch genes on or off, and those changes can sometimes be passed down too. So it's not just the letters in your DNA, but also the punctuation, if that makes sense. Oh, and I have to tell you, last month I was at the Natural History Museum, and I found myself staring at this display of skeletons. A mouse, a bat, a human, and a whale. And it just hit me. Despite how different we look, we all share so much of the same genetic code. I mean, Hox genes, for example, control body plans in everything from fruit flies to us. It's humbling, isn't it? To realise we're all riding the same genetic highway, just in different vehicles. So, let's zoom in a bit. If DNA is the blueprint, then cells are the builders. Cell theory says all life is made of cells, and every cell comes from another cell. It's like this endless relay race, passing the baton from one generation to the next. Mitosis is the process that keeps things ticking, making identical copies for growth and repair. And then there's meiosis, which is a bit more creative, mixing up genes for sexual reproduction. That's where you get variation, which is, kind of the spice of life. And think about this. Every human starts as a single cell, a zygote. From there, through mitosis, you end up with, about 37 trillion cells as an adult. I always lose count after a few billion, to be honest. But it's not just about numbers. Embryogenesis is this mind-blowing choreography, with stem cells dividing and specializing, and epigenetic marks guiding the whole process. It's like a dance, but with way more at stake than just stepping on someone's toes. Speaking of choreography, let's talk about Dolly the sheep. Remember her? She was the first mammal cloned from an adult cell back in 1996. Scientists took a cell from an adult sheep, reset it to an embryonic state, and voila, a new sheep, genetically identical to the donor. It was a huge deal, not just for science fiction fans, but for anyone interested in what makes an individual unique. Dolly showed us that you can copy the genetic material, but the environment, the epigenetics, the little quirks, they all matter too. So, continuity isn't just about making a carbon copy. It's about the interplay between sameness and difference. Now, let's pull back and look at the bigger picture, the webs of life that keep everything connected. In ecosystems, it's all about interactions. Food webs, keystone species, the whole lot. If you take out a keystone species, the whole system can wobble or even collapse. There's this constant cycling of energy and matter, producers, consumers, decomposers, all playing their part. Only about 10% of energy gets passed up each trophic level. So there's a real efficiency game going on. But here's where things get really interesting. Technology is starting to reshape how we think about life's continuity. Cryopreservation lets us freeze cells and embryos, pausing life in a way that would have sounded like science fiction not long ago. And then there's CRISPR-Cas9, which is, well, basically a pair of molecular scissors for editing genes. It's precise, it's powerful, and it's raising all sorts of questions. Are we reinforcing the chain of life by fixing genetic diseases? Or are we, maybe, challenging what it means for that chain to be unbroken? I'm not sure there's a simple answer. On one hand, genome editing could help preserve endangered species or improve crops. On the other, it blurs the line between natural inheritance and human intervention. So, as we look to the future, the continuity of life is still, well, unbroken. But it's also evolving, shaped by both nature and technology. That's all for today's episode of Continuity of Life. Thanks for joining me, and I hope you'll tune in next time as we keep exploring the threads that tie us all together. Until then, take care and keep wondering.